That's me. Hello, I'm Arda. I'm a third year postdoc at the Genomic Sciences and Systems Biology department. So I'm a system biologist by training. Most of my research is focused on methods and application rather than like question driven. And lots of the tools or models that I use can be like encapsulated around this representation learning toolkit, which is essentially taking multimodal data sets and transforming those into a, let's say, machine readable or visualization wise useful reductions so that downstream analysis could be performed. And I've put like 4 projects that I'm like actively working on. One of which is this some morphology to mutation mapping. Basically, it's kind of like a predictive multi-instance learning problem where we have a bunch of cell images from a patient. We are trying to predict whether we can identify certain mutations or not. And that representation zoom out looking plot is fully unsupervised representation of what the model has learned so far.
Oh, is that actually from that project?
Yes.
Oh, cool.
Going to show like tomorrow.
Yeah, I haven't seen it. Cool.
But it's like quite interesting to see that like different cell types are being clustered so that we can be very much like sure that the model is like learning something useful. And the question we are asking during the training is basically if you corrupt the image, can we reconstruct the original image, which like the middle column is it corrupted, like random noise, random patches, and then the right column is the reconstructed image. Although it's like a bit blurry, the overall like morphology of the cells are being captured. So this model is going to be like fine-tuned and training more, but it seems like it's doing its job so far.
That's pretty cool. So cool. That's a brand new project. I haven't seen that yet.
And then the second project is in collaboration with Berkeley Lab, is the bottom left figure, which is a temporal characterization of transcriptional changes and collateral drug responses in osteosarcoma data set. And it's just like a simple representation of, again, a latent model that we train, which is able to capture the treatment and then the control axes of the evolutionary process. So that's pretty much we're trying to figure out like what functional pathways are happening along the evolutionary axis. And then the third project, which is kind of popping up from time to time with different aspects, but it's pretty much like related to explaining the machine learning AI models to be applicable in clinical settings. Because the issue is, even if the predictions are good, the reason that the model is making those predictions are kind of like black box, we don't know. So what I'm trying to see if we can use these like representation learning toolkits to address that question. And 2 figures, again, like isometric embeddings, the left-hand side for expression, right-hand side for mutation. Again, this is like a multimodal and tissue cell type agnostic model. And the higher, the brighter the color is like drug resistant, and then the drug sensitive, and then different patches of drug resistance are basically because of different mechanisms, or because of different transcriptional changes. So we can see that for the same drug, the model is able to capture distinct resistance patterns, and we are able to go back and ask the question of what are we extracting, why are we making these predictions. So it's kind of like in progress, but it seems to be an okay model. And then the final one is a bit distinct, which is like generative modeling of amino acid sequences. And this project popped up from a collaboration trying to understand whether we can model TCRs, peptides, and MHC interactions from solely sequence analysis perspective. So we are applying a standard like LLMs, different types of transformer-based models with different training strategies to see if we can both predict those interactions or generate de novo sequences. So given the TCR sequence and then the MHC context, can we generate like peptide sequences out of blue that we know that will interact with the TCR or not? So